All right, good morning, everybody. I hope your spring day was delightful. Um, they used to have 2 reading days before finals, and now they only have one. So guess where your spring day came from? Yeah, anyway, well, hope it was nice. As you know, you have a test that opens tomorrow. I've already written it, and it's 20 questions long. I don't know how hard or difficult it is, so you'll just have to see. And I think that's all I have to say about that. Okay, so today I'm going to tell you an interesting story of how chemistry, and in particular organic chemistry, revealed some things about how DNA replication works, at least in one particular DNA polymerase. So we won't go into this a lot, but as you may have learned, there are enzymes called DNA polymerases that start at the 5' end of DNA and continue synthesizing from 5' to 3'. And yeah, that enzyme is called a DNA polymerase. And of course, it's good at putting in the complementary base pair sequence as it synthesizes along. So, for example, if we had a sequence T, A, C, and G, the polymerase would be moving along, and on the other side would put the complementary. For G, that would be C, for C, that would be G, for A, that would be T, and for T, that would be A, and so on. And this is how the information in DNA is propagated. Now, we've learned about Watson-Crick hydrogen bonding. You guys know that the GC pairs have 3 hydrogen bonds, the TA pairs have 2 hydrogen bonds. And it's a natural, we've shown how that hydrogen bonding pattern is complementary. So it's a natural thing to assume that what the polymerase is doing when it installs the correct nucleotide opposite its partner in the coding strand, or in the strand that's already there, it's easy to assume that the polymerase is just matching up hydrogen bonds. And that the reason that, for example, the polymerase incorporates C opposite G is because they have a complementary hydrogen bonding pattern. That sounds reasonable, and for a long time that was the assumption for how things worked. So let me show you some data in terms of the stability of some various DNA duplexes where we're going to vary the nucleotide that gets incorporated at this position X. And we're going to see in particular what happens when you have mismatches. All right, so I've written out a sequence from 5' to 3', and then the opposite strand from 5' to 3', we would write in the complementary sequence. And this is going to be sort of this central pair. As I said, we're going to vary what, we're going to hold X constant and vary what Y is, and see what that does to the stability of a double-stranded DNA duplex. And then we'll talk about the polymerase. But right now we're just worrying about this double-stranded DNA. And you can measure the stability of a DNA duplex, and the experiment to do this is actually really straightforward. Double-stranded DNA has a different absorbance at 260 than single-stranded DNA. That's because 260 nanometers, by the way, that's in the UV. The reason that the absorbance is different is because in the DNA duplex, well, the thing that absorbs at 260 is those purine and pyrimidine bases. and their environment is dramatically different in the DNA duplex than it is in single-stranded DNA. Remember, the duplex are hydrogen-bound and they're stacked on one another. So you can actually measure the stability of a DNA duplex by monitoring absorbance at 260 nanometers and then turning up the temperature and watching the DNA, the signal at 260 nanometers go away in this sort of sigmoidal fashion. And the midpoint of the transition we're going to call a melting temperature, or TM. And this will be sort of a measure of stability. So if the DNA duplex, if you have to turn up the temperature higher before the duplex dissociates, and you see that drop in absorbance, then the DNA duplex is more stable. All right, so let's suppose that we keep X constant. We install thymine at the X position. And then we're going to actually make a complementary strand. And we'll talk later today about how you synthesize DNA. It's actually really easy. So easy that robots do it. And if today you decided you wanted a particular sequence of DNA, either single or double-stranded, you could go on any number of websites, order it, and it would be to you either this afternoon or early tomorrow morning. It's like really fast. So we can do this chemically. Now let's suppose that we make we make 4 different versions of this second oligonucleotide, where we let Y, the Y position vary between A, G, C, and T. So if we let Y equal A, that's a match to the thymine, right? But everything else is a mismatch. And let's see how the melting temperature varies. You would expect that the most stable duplex would be the one that has the match, and indeed that's the case. If you put, if at the X position you have thymine and then you put adenine opposite thymine, the melting temperature is 42 degrees Celsius. I know that somebody in the audience is currently asking, do I need to know for the test that melting temperature is 42 degrees Celsius? No, please do not waste any time memorizing that. We're not about memorizing strange numbers. Okay, so not strange numbers, sure, we can memorize, but strange numbers, just kidding, sorry, lame. Let's see. So if you put G opposite the T, that's a mismatch, and the melting temperature goes down. And actually you can infer, this is just me geeking out on biophysics, you can infer from the melting temperature a change in free energy of association of those 2 helices with each other. And so if we do that, we'll call the mismatch sequence sort of our starting comparison point of 0, and then everything else, if it's less stable, the difference in free energy should be positive. So with the mismatched pair, it's a loss of 2.2 kcals per mole of stability. Okay, well, so far that tracks with what we believe in terms of how the DNA polymerase might work, and we might say, well, the DNA polymerase could recognize this mismatch because the resulting DNA duplex is much less stable if you have a mismatch. And that is true as well for other mismatches. So, for example, the C and A mismatch, the C mismatch, putting C opposite T gives you a melting temperature of 29, and it's less stable than the matched pair by 3 kilocalories per mole. And then the T mismatch, T versus T mismatch, is about as bad as the T versus C mismatch, and about the same difference in stability in terms of free energy. So the take-home message from these data are that the DNA duplex is more stable when you have matched pairs. Surprise, surprise, okay? But the question we haven't answered, we've only been talking about the stability of this double-stranded DNA segment. Now the question we want to answer is, does the DNA polymerase use hydrogen bonding as a way to recognize whether you're going to have a match versus a mismatch? You see how those are two different questions? You're asking about the stability of the DNA duplex versus how the enzyme that makes the DNA puts the right nucleotide at the correct position. All right, so chemists can ask this question because we can design a nucleotide that has any number of features. We're not limited to the 4 bases that nature uses. So I'm going to redraw thymine here for you so that you can see the hydrogen bonding pairs or the groups that are involved in hydrogen bonding that we've been talking about. So this is the thymine base. And chemists at Stanford in the lab of Professor Eric Kuhl, he's got an awesome last name, but it's spelled with a K, asked the question, what if we replaced thymine with something that couldn't hydrogen bond? And I want, it's fine. What did we do? Oh, I erased the Kuhl. That's funny. I want to pull from our lecture last time just an image of the hydrogen-bound pair of thymine with adenine. Okay, so the students in Professor Kuhl's lab asked the question, what if we replaced these groups, the oxygen on the carbonyl oxygen, the NH group, and this carbonyl oxygen with something that was about the same shape, but nevertheless was much poorer at hydrogen bonding. So they invented a nucleotide which they called DF for difluoro, and their nucleotide looked like this. So I'm not going to draw the sugar, and actually this is cool chemistry because instead of cool chemistry, see what I did there without even intending to? And the puns just write themselves. They created what's called a non-hydrogen bonding isosteer. Isosteer means same shape, same size, but less capable of hydrogen bonding. And in contrast to thymine, thymine is attached to the sugar via the nitrogen. It's an N-glycoside. Here they used a carbon. I'm not sure why, maybe for ease of synthesis. It's a C-glycoside. They used an aromatic ring where they put 2 fluorines in place of the 2 oxygens, and then just a carbon-hydrogen bond here in place of a nitrogen-hydrogen bond. So whereas before you had hydrogen bond acceptor, hydrogen bond donor, and hydrogen bond acceptor, here now you have fluorine, which is electronegative and does have lone pairs, but because it's more electronegative than oxygen, it's going to be less likely to share those lone pairs with a hydrogen bond donor. So there are 2 fluorines there, and then you have this carbon-hydrogen bond, which is way less polarized than a nitrogen-hydrogen bond, and so should be a much worse donor. So we've basically designed something that takes up the same amount of space as does thymine, but lacks the hydrogen bonding capability. Okay? Makes sense? This is an example of what chemists do. It's called structure-activity relationships. People do it in medicinal chemists do it in the pharmaceutical industry to try to optimize compounds. This is something we think about a lot in organic chemistry. We do this in my lab all the time when we want to determine, is that particular functional group important? We replace it with something else and make a change. So DF is the non-hydrogen bonding version of T. Okay, so let's ask the question in terms of duplex stability. What happens when you incorporate when you incorporate F, DF, the nucleotide, instead of T? All right, so we got the same sequence. And we're going to, at the X position, we're going to put this DF nucleotide that doesn't have hydrogen bonding ability. And then we're going to then we're going to measure the stability of the DNA duplex when Y equals, again, A, G, C, and T. And if we measure the melting temperature in degrees Celsius, what we see is that the duplex is way less stable. The duplex is way less stable when you have DF at the X position than when you have T at the X position. Realize we're comparing a lot of things here, so I'm going to pull these data down just so you can see. Here was when where the X position that we're varying was thymine, and here is when it's this non-hydrogen bound isosteer. You know what would be really great? There we go. Maybe that's easier to compare. So notice that when thymine is at the X position, there's a large difference in stability between the matched pair versus the mismatched pair. Okay? But when the non-hydrogen bonding isosteer DF occupies that F position, there's almost no difference. It makes no difference in terms of the stability of the double helix, what you have opposite DF. Okay, so conclusion number one is that hydrogen bonding is crucial for duplex stability, for DNA duplex stability. And this is well known, so this is kind of a gee whiz, you've just shown us the same thing everybody already knew result, but it gives you a sense for the fact that if you don't have the hydrogen bonds present, the stability gets a lot lower. And then, moreover, in terms of stability, this DF non-hydrogen bound base seems to be a lot less discerning in terms of its partners than thymine is. Right? Do you see how it doesn't matter what the partner is if we use the difluorotoluene base, but if we use the thymine base, it matters a lot who the partner is? Okay. So now, that answers the question in terms of what makes a DNA duplex stable. And to this day, actually, if you know what the sequence of your duplex DNA is, you can predict its melting temperature pretty well. And sequences that have more GC pairs are way more stable than sequences that have more AT pairs, because there's 3 hydrogen bonds in a GC pair and only 2 in an AT pair. All right, so the next thing that they did in the Cool lab was they said, okay, let's now use the top strand here with X. And let's see what gets incorporated by a DNA polymerase when this X position is T versus when the X position is this non-hydrogen bonding isosteer, DF. All right, so, oh, great. I should really learn to charge my Apple Pencil. All right, so you have this enzyme, the DNA polymerase, and actually this was a fragment of a DNA polymerase called the Klenau fragment. That's not important to anybody unless you're into DNA polymerases, but you'll hear that from time to time in a biochemistry class. So they took this particular DNA polymerase, they fed it this template strand, and then they asked what gets incorporated opposite. Okay? Now, the hypothesis is if the polymerase was using the hydrogen bonding pattern to incorporate the correct base opposite T, then you would predict if you remove the hydrogen bonding pattern, the polymerase should be terrible at incorporating the correct base at this position, right? The result, by the way, I mean, non-surprising, is sure, the DNA polymerase puts A opposite T. But the question is, what is it going to put opposite the non-hydrogen bonding isosteer? And if hydrogen bonding mattered, if the polymerase was using the hydrogen bonding pattern and the stability of the resulting DNA duplex, you would predict that if you take the hydrogen bonding away, there'd be no selectivity here, right? But it turns out that the polymerase, surprisingly, puts the right base there, which was kind of one of these mind-blowing results because, and of course there are percentages here, and I haven't written down what the percentages were, but by far the major base that the polymerase incorporated opposite this thing that cannot hydrogen bond was the correct base. So the question is, how on earth did that happen? And the conclusion seems to be that this particular DNA polymerase doesn't care so much about the hydrogen bonding. That's important for duplex stability. But instead, the polymerase uses shape rather than hydrogen bonding to put the correct base in the correct spot. So conclusion number 2 is that for the particular DNA polymerase, shape is more important than hydrogen bonding for selectivity. And that's really kind of cool, and it's cool because it's counterintuitive. It's really easy to look at the DNA structure and to predict, oh yeah, well, the polymerase knows the right one to put in because of the hydrogen bonding pattern. And it's kind of mind-blowing to realize that actually the polymerase doesn't care that much about the hydrogen bonding pattern. It cares more about the shape. The idea here then is that if you have the wrong base opposite the template strand, if you have a mismatch, somehow that mismatch pair doesn't fit as well in the active site of the enzyme. And there are a lot of crystal structures of DNA polymerases with strands of DNA that are in the process of being synthesized. So here happens to be a DNA polymerase. Here's the PDB code. Actually, if you want to see this yourself and go look at it, you can just type that into the protein data bank and you'll get the coordinates for this structure and you can visualize it. Here is a matched pair between a G on a coding strand or a template strand and then a C, and this is a this is this molecule is a mimic of CTP. Sometimes when you want to do crystallography of enzymes, you can't put in the actual thing because the enzyme will instead just do chemistry, and so they put in a mimic of cytidine. This, you'll notice, isn't actually a sugar, but it fits in the active site in the right spot. And here is the 3' end of the strand that the polymerase is making, and then here is where the next incoming nucleophile would be. Here is the cytosine base, and then here is the guanine base, and you can see the hydrogen bonding, and you can see that they fit in the active site in a somewhat flat orientation. Now, if you take the same, a related situation, and then and look at a mismatch, what you see is instead of the nice sort of planar pair, in a mismatch between thymine and guanine, you're starting to see some steric clashes. You see how those bases are tilted out of planarity? That's because they're bumping into each other, and the polymerase can sense that and know to put the right thing in the right place. All right, questions about that little story? Yeah. But this is how organic chemists make conclusions. We test, we make molecules that vary from the natural ones, and we see what the consequences are. And based on that, we can draw conclusions about how biology works. Okay, there's a whole field in chemistry out there about expanding the number of base pairs that DNA can have. If you're interested in that, you can look up not only the stuff in the Cool lab, but also from Floyd Romsberg's lab at the Scripps Research Institute. He's generated some alternative DNA base pairs that the idea is long term, could you expand the genetic code by having some more options? Anyway, okay. So now I want to talk about how oligonucleotides are synthesized. As I said, everything we just talked about would not have been possible without the ability to make small pieces of DNA. And a lot of modern chemical biology is related to being able to make the right pieces of DNA. So we want to talk about how this can happen. I'm not going to mention in class or on the test really the PCR reaction, though you read it as part of your study guide presumably. Be aware of it and know what it is, but we won't talk about it in a lot of detail. Though it is important, right? A lot of you have had PCR-based COVID tests, and so it's useful to know what that is. So how do you make an oligonucleotide chemically in the lab? The answer is on solid phase. And so I'm going to show you how this works. You have some kind of resin, and then there's a linker between the resin, and we're not going to say what that linker is. There's a linker between the resin and the 3' end of DNA. So here is your ribose sugar, and you've got a particular base at this position, and we're not going to draw what the base is. We'll just say that's base 1. And then on the 5' end, you've got a protecting group. And the idea here, the reason you need protecting groups is because you have in a nucleoside, you have two different OH groups that you need to distinguish between. All right, so the protecting group that's on the resin for synthesis of nucleotides, or the protecting group that is used for the 5' hydroxyl group, is called dimethoxytrityl. What this is it's 3 benzene rings attached to the same carbon, and 2 of the benzene rings have electron donating groups at the para position. So all of that, this is a new example of a protecting group for an alcohol. You've seen silyl protecting groups for alcohols before. This is a new one called dimethoxytritol. And we can use that abbreviation. And the idea is that it prevents this oxygen from being the nucleophile until we're ready. All right. So you remember in solid phase peptide synthesis how we had a deprotection step for the amine, and then we had a coupling step. We're going to do the same sorts of things here. We want to use the 5' OH group as a nucleophile, and so we're going to bring in the next nucleotide and make a new bond there. So first we have to unmask that dimethoxytritol group. So the way you do this is with a strong organic acid, and the acid of choice is trichloroacetic acid. And this is a liquid, so you can do this reaction just by adding liquid trichloroacetic acid. And what happens in this reaction is you protonate the OH group, you protonate the 5' oxygen. That turns this part of the molecule into a good leaving group, and then it picks up and leaves. And what you're left with, the thing that leaves, or rather, you're left with, oops, got that wrong, a carbocation. And the carbocation is resonance stabilized. So this is basically an SN1 reaction. And that removes the protecting group on your nucleotide and exposes the 5' hydroxyl group, which you can then use as a nucleophile. The neat thing about this tritol cation is it's resonance stabilized, but it's also very colorful. It's a bright orange. So you can actually monitor this reaction just by looking at the color to determine whether or not it actually happened. And most oligonucleotide synthesis instruments now monitor absorbance wherever this cation absorbs in order to determine whether this reaction was successful. All right, so that's a deprotection step. Now, like with solid phase peptide synthesis, we need a coupling step. And this chemistry is a little bit new. So we're going to use the 5' hydroxyl as a nucleophile. But remember that when we make a nucleotide, we want one sugar attached to the other through a phosphate linkage. We haven't done anything really in this class with phosphorus chemistry. So what we're going to talk about now is it's not going to make a huge amount of sense in terms of why it has to be this way. But I want to show you the chemistry because it's so highly efficient and it's enabled so much work in biology. So the next nucleotide we bring in with the 5' group protected as the dimethoxytritol. I'm going to abbreviate that as DMT. Then here's the 3' oxygen, and that's going to be bonded to a phosphorus. And the phosphorus is going to be also bonded to another oxygen and a nitrogen. And this is called a phosphoramidite. You don't need to remember that, but you might hear people talking in the future about phosphoramidite chemistry. Our goal here is going to be to have the oxygen attack as a nucleophile to replace this nitrogen group on the phosphorus. Okay? The problem is, if we want to use that as a leaving group, it would leave as this. Anybody recognize that? You saw it before at the beginning of the semester. What is it? It's a really strong base. That's LDA if we have the lithium, right? So really exceedingly cruddy leaving group. So to make the reaction happen, we're going to need an acid to protonate this nitrogen, but we can't have it be too acidic because otherwise we'll get rid of the dimethoxytrityl group. So they had to tune this very carefully to get the right kind of acid. And so the thing that works is the following molecule. It's a heterocycle, and a really complicated one because you've got 4 nitrogens in it. It's called tetrazole, and to make matters worse, they also have a sulfur there. All right, so this is a tetrazole. This molecule is called a tetrazole. Its role is as an acid and then also as a nucleophilic catalyst. Both an acid catalyst and a nucleophilic catalyst. So step 1 is for the lone pairs on the nitrogen to pull off the proton on tetrazole. And it's not immediately clear whether the pKa's should work out, at least based on things that we've talked about. So you're just going to have to accept that the pKa's are such that that reaction works fine. So protonating the nitrogen on the phosphoramidite should convert it from something that was a poor leaving group to something that's a okay leaving group. Oh, dang. There we go. But there's evidence that before the OH group on the nucleophile attacks, there's evidence that the conjugate base of the tetrazole that we just talked about, oops, that's a nitrogen there, negative charge here, sulfur, ethyl group. There's evidence that that tetrazole actually acts as a nucleophile to kick off the nitrogen. And there's a lot of examples of this kind of nucleophilic catalysis in nature, in chemistry. I don't know that we've talked about any of them in a lot of detail, but the idea is that the tetrazole is a good enough nucleophile to kick off the amino group, but itself is a decent leaving group. And I think you can see how that might be the case simply because, well, right, so there's evidence for this kind of species, and the idea is that this would be an even better leaving group than the nitrogen. So I don't know what we're going to do for that. Maybe we'll make it darker green for even more go. And at that stage, then, our 5' hydroxyl can act as a nucleophile and kick off the tetrazole. And what we get after that is our 2 nucleotides bonded together. So here is the resin. Here is the first base. Here's the 5' hydroxyl group. And then that is now bonded to the phosphorus. A lot of drawing here. Okay, so the new bond we made is right here. And that's linked the two nucleotides together. So we can continue onward by deprotecting the DMT and then bringing in another phosphoramidite and doing this coupling reaction. But if you think about where we need to end up, this linkage is not quite the phosphodiester linkage that we're looking for. This is not the phosphodiester linkage you're looking for. Lame. So we're looking for this, and we have phosphorus is not quite at the right oxidation state. But on the other hand, we don't want it to be this because you've got a negatively charged oxygen on the phosphate ester, phosphodiester, and that might interfere as a nucleophile in the coupling step. So that's why we've got this weird group on the phosphorus. But at this stage, what we do is we oxidize this molecule to get one more oxygen on that phosphorus. So let's copy that, or not. Actually, let's just try to draw it neater. So here's the second base. Here's the dimethoxytrityl group. Here's this other base, and then here is the resin. All right, so we're going to use iodine and water to install an oxygen on this phosphorus. So the phosphorus is going to actually act as a nucleophile. I'm not going to show the mechanism here, though basically what happens is we make a new phosphorus iodide bond, and then water attacks that to replace the iodide with an oxygen. Okay. And then actually at this stage, so that was step 3, this is the oxidation phase. And then you can repeat steps 1 through 3 hundreds of times to get a really, really long oligonucleotide. There's another step that's typically included, but we're not going to worry about it. It's called a capping step, but it's more about efficiency than it is about anything critical happening. And let's see. I feel like at some point we lost an oxygen, didn't we? All right. So you can see that by repeating steps 1 through 3, we could have a very, very long oligonucleotide. The last step is going to be to cleave from the resin, and we also are going to need to remove this group to liberate a negatively charged oxygen. All right. But before I show you that, question so far about this, I'm guessing we're looking at it and thinking, why the heck? Why the heck are you doing this to us? But yeah, go ahead.
So I remember from the protein synthesis that acid would cleave the linker bond with the resin.
Right.
So why doesn't that happen here?
Why doesn't that happen here? Yeah, the question is, when we did peptide synthesis, we used an acid labile linker. So this linker must be stable to the acidic conditions for deprotecting the trityl group. And it also has to be stable to the tetrazole conditions. And it also has to be stable to the oxidizing conditions, the oxidation step. So yeah, it's just the chemistry here, and I haven't talked about it. I would need to look it up. I actually don't know what it is. But the bond that links the nucleotide to the resin is stable to acid, so it won't fall off. And that just has to do with the functional groups that are present there. It will turn out that what removes this from the resin is the same thing that will, when we're finally done, will also remove this group. Yeah? What makes the tetrazole a good leaving group? It must have to do with the fact that this is the conjugate base of a decent acid. I mean, that's what I basically just did. Is I told you it's a good leaving group because it's a good leaving group. I guess I don't have a better answer than that. I think you could say that the negative charge is resonance stabilized because I could move this lone pair around the ring, and then that would put a negative charge on that nitrogen, and so it's probably resonance stabilization. Okay, what else? All right. So once we've got our long oligonucleotide, and so what I would do at this point is I'm just going to put another R here, and this means many other nucleotides. Then in a final step, the final step is cleaving and backbone, phosphate backbone deprotection. This involves ammonia, and ammonia will break this bond between the resin and the oligonucleotide, and it also will do an elimination reaction where we remove this proton and the negatively charged oxygen leaves. And that then establishes our negatively charged phosphodiester linkage. And that will give us our free oligonucleotide. And the cool thing is that in the time that we've been talking about it, we could have done this reaction, done these reactions to put an additional nucleotide on our chain several times because the chemistry is quite fast. Okay, so therefore what? This is another example of solid phase chemistry, and the idea here is that because the molecule is immobilized on solid phase, you can rinse away byproducts. You don't have to do purification. There's some new chemistry here. The first step was an SN2 reaction. This chemistry, phosphoramidite, was sort of new. The oxidation was sort of new, but that is sort of how it works. Now, I guess I can relieve you by saying I did not ask a question about this on the test. I might do it in the future, but I wanted you to know how people make oligonucleotides and how you can order them. All right. Sorry. That's enough. Have a good day.